My name's Charles Mulligan. I'm a member or professor in the Department of Pathology at St. Jude Children's Research Hospital in Memphis, Tennessee. We know a great deal about how ALL or acute lymphoblastic leukemia develops. In essence, this is a genetic disease that is one or more genetic changes to the normal DNA makeup of a cell, are acquired in a developing white blood cell, and this causes them to not develop properly and to proliferate uncontrollably. These genetic factors can be either inherited and acquired, that is, they can be present at the time of birth, or they can be picked up over time during life. We know now that probably 10 to 15% of cases of acute lymphoblastic leukemia have some level of genetic predisposition. That is something already present in the genetic makeup of an individual that predisposes them to develop a tumour or ALL. It's possible that this is an underestimate. We're still not fully aware of all the genetic changes that may predispose someone to develop ALL, but again, a substantial proportion of individuals, particularly children, are predisposed to develop the disease. ALL is particularly common in some situations, an example of this being Down syndrome, which is associated with a very high risk of developing acute myeloid leukemia, but also acute lymphoblastic leukemia. Now, apart from that, we know that the majority of cases of acute lymphoblastic leukemia have quite distinct genetic changes. If one looks at ALL and compares it to other tumours, for example, tumours that are common in adults and associated with environmental exposures such as sunlight with melanoma, or smoking and lung cancer, or diet and colorectal cancer, the absolute number of genetic changes in ALL is relatively low. Often it's only a single digit number, or 10 to 20 genetic changes in total. However, those genetic changes are often highly important. They target key cell growth, differentiation and control pathways, and they have a direct role in contributing to disease. The genetic changes are varied in type. They can be single letter changes of the DNA code. They can be more gross changes where there are breaks in chromosomes, and those chromosomes can rearrange in abnormal ways, and that can disrupt the function of normal genes and cause them to function in new and abnormal ways. And again, that contributes to the development of ALL. Often this process takes time to develop. There are some subtypes of leukemia where we know that the first genetic changes are acquired very early in life, and they can be detected in cord blood at birth, but only a proportion of those individuals go on to develop the disease. And when it does develop, it takes some years. And together, those observations indicate that there is a series of genetic changes that are acquired over a period of time that are required for ALL to develop completely. Sometimes that's not the case. Sometimes ALL develops very early in life, for example, in infancy, and it's in that subtype of leukemia, only a single genetic change can be sufficient to develop the disease. It's often asked if there are environmental exposures that contribute to the development of ALL. And it is recognised that there are some that contribute, but it's likely that these have a low influence in the majority of ALL cases. For example, things like industrial exposures, benzene, ionising radiation, and so forth. These have been linked to cases or clusters of ALL, but again, they likely account for only a small minority of cases. We don't fully understand why all the genetic changes that develop are acquired. It's likely that some of them are related to a normal burst of proliferation of lymphoid cells that occurs normally very early in life, that's associated with the normal development of the immune response and the maturation of that response that enables children to respond to normal infections, bacteria and viruses. And sometimes that process can trigger changes in genes that shouldn't occur, that then are conferring an advantage to the cell. They're preserved, and that's a step that leads to the subsequent development of fully fledged ALL. What drives ALL development in older individuals, because this is a disease that occurs right across the age spectrum, is not fully understood. We do know that the genetic subtypes of ALL differ distinctly with age. And there are certain subtypes of ALL in children that are associated with very favourable outcome that are rare or absent in adults. And conversely, there are some subtypes that are often associated with a high risk of relapse that are uncommon in children, but much more common in adults. Why they are acquired preferentially at a certain age, we still have a limited understanding, but we do have a good understanding of those genetic changes and increasingly how they're contributing to tumour response. And excitingly for the field, these are giving us new insights and approaches and opportunities to selectively develop therapeutic approaches that are more effective for many of these high-risk subtypes of leukemia.